So the iGEM startup program helps iGEMers turn their ideas into real biotech ventures. Through the Venture Creation Lab, teams sharpen their vision, refine their business models, and get ready to launch. The Startup Showcase is where all that hard work comes to life. It is a stage for the most promising iGEM founders to pitch their startup ideas to investors, industry leaders, and the global bioinnovation community.
And so now, let's meet the teams who've made it here.
Hello, iGEM. My name, thank you guys. My name is Kenzo Salazar, and we are Pomegranate Biomanufacturing, making the biomanufacturing of volatile enzymes possible. Proteases are a class of enzymes which cut peptides. This mechanism of action is highly sought after, making proteases account for 60% of the global industrial enzyme market. And the field shows no sign of stopping, with an expected doubling of the market size by 2035. The mechanism of action, however, is highly volatile, meaning many proven proteases cannot be manufactured at scale recombinantly, leading to significant losses of around $1.2 billion per year. We at PomBio have the solution and intend to capitalize on the market with our novel manufacturing mechanism. The status quo of biomanufacturing is cytoplasmic or secretion-targeted protein expression. High concentrations of heterologous proteins in non-optimal environments leads to irreversible aggregation, causing functional product loss and cell death. As mentioned earlier, proteases cut, destroying native peptides, limiting protease production to low levels. Finally, secretion is convoluted, with many opportunities for error and dozens of chaperones necessary. We localize manufacturing in the vacuole of yeast cells, specifically lowering cellular burden, tuning the environment to stop aggregation, and preventing proteolytic cleavage, allowing for large-scale recombinant manufacturing of volatile enzymes. The components of our solution have already been individually validated, with us now working on combining the positive results in snake protease manufacturing, yeast vacuole tunability, and vacuole targeting, already with our own positive lab results. With hemorrhage taking life every 9 minutes in the United States and the global blood diagnostics market being $90 billion, we are first focused on batrasobin, a hemorrhage diagnostic currently sourced from snake venom, projecting a reduction in production cost from $90 to just 21 cents. The valuation of our serviceable market is $1.5 billion, and we expect to capture 10% of this market within the next 5 years. We expect to have MVP within the next 2 years, shifting our focus to commercial development, regulatory approvals, and distribution in the 1 to 2 years afterwards. Because there are currently no method or utility patents in the field, we have begun the IP process with Princeton University's legal team, giving us initial control of the market. We will employ a twofold B2B business model. For each protease we develop, we will set up in-house manufacturing while selling off the appropriate diagnostics division to larger in vitro diagnostics companies, leveraging their greater resources in hospital settings and giving us initial capital for further R&D, while we continue to generate revenue by providing the reagents necessary. My apologies. This twofold business model allows us, sorry about that. This twofold business model allows us to control low-cost recombination while profiting off the high margins of the diagnostic market. I myself focus on biomanufacturing at Princeton University, with my co-founder, Arif Ansari, focusing on finance, therefore leading the market and business strategy division. We have recently partnered up with the Averra research group at Princeton University. This research group specifically focuses on vacuole yeast biomanufacturing in organelles, such as the production of biofuels in the mitochondria, already with several spin-off technologies on the market. We are currently looking for clinical and biomanufacturing industry advisors to join our team. But with our team of mentors, professors, and clinical advisors, we are already ready to bridge the gap between protease manufacturing and in vitro diagnostics companies. I would like to end off with a story. In 1978, Eli Lilly changed the world by producing the first commercial peptide pharmaceutical, insulin. This paved the way for the revolution that we have had in peptide pharmaceuticals, from psoriasis treatments to GLP-1 agonists. They showed that by perfecting a technology, you become an industry leader in that field. We now have the technology to do the same. By perfecting volatile enzyme biomanufacturing, we at Pomegranate Biomanufacturing will become industry leaders in our field. Thank you.
Thank you.
Every minute counts when someone is bleeding out. In cases of severe traumatic blood loss, irreversible damage can begin in as little as 5 minutes. If the average emergency response time is 8 to 10, which can easily increase to half an hour in remote areas. Most critically, ambulances don't carry blood. Each year, around 2 million lives are lost due to hemorrhage, many of these deaths preventable with timely transfusions. Except, whole blood is not shelf stable. It expires in 42 days under constant cold storage and just 4 hours at room temperature. Combined with the ongoing blood shortage crisis, in which less than 3% of eligible North Americans are donating blood, our current system is fragile. For decades, we have tried to replicate blood's life-saving oxygen-delivering functions through hemoglobin-based oxygen carriers. But research has been largely limited to mammalian hemoglobin, which readily dissociates outside of red blood cells, causing toxicity.
So why brute force mammalian hemoglobin to meet our needs when we can rely on the natural design that has been refined for millions of years of evolution? Certain invertebrates don't have blood cells. They have hemoglobin molecules that are specifically designed to transport oxygen stably in the bloodstream and flow freely. So stable, in fact, that instead of 42 days, we can enhance the shelf life to up to 2 years without the need for cold storage. This allows us to retain the oxygen-binding power of human hemoglobin without the instability that has limited so many past attempts. Using well-studied recombinant methods, we are developing the means to scalably produce this biomolecule for human therapeutics. We've taken the recently characterized protein sequence and modified it for commercial production in a yeast expression system. Unlike traditional farming methods, this allows us to tailor our sequence for enhanced yield and enhanced therapeutic potential.
The global market for oxygen therapeutics is expected to exceed $1.5 billion by 2030. Yet, an easily modifiable, cost-effective, and universally compatible oxygen carrier still does not exist. That gap is exactly where Synferra positions itself. Our financial modeling shows that at scale, Synferra can achieve near 100% margins and price points that are competitive with existing hemoglobin-based oxygen carriers. Furthermore, our commercial pathway is designed to generate early revenue and avoid regulatory friction by leveraging our protein's applications beyond just therapeutics. Take cell culturing or bioreactor oxygenation. These are industrial and research markets that we intend to target first, where the barrier to entry is low and validation time is fast. For that same reason, we'll move on to veterinary application, using that traction to fund and de-risk our entry into multi-stage clinical trials and eventual human therapeutic use. This is our team, advisors, and partners that have made our company possible. Every minute counts, and for too long, we've accepted that those minutes are out of our control. Synferra is here to change that.
Thank you, Synferra. Congratulations again.
Hi everyone, we're Averra.
Averra.
And we believe that the future of farming lies beneath our feet. We deliver real-time soil intelligence for precision agriculture. Soil isn't just dirt, it is one of our planet's most powerful natural engines, capable of climate resilience, food security, and now economic opportunity. With the rising carbon market, soil is no longer just seen as dirt, but it is now recognized as a living asset, one of tradable and measurable value alike. But here's the problem. Without data to understand the soil, you can't unlock its potential or the value that comes with it.
And existing soil testing methods haven't kept the pace with this innovation. We spoke to farmers directly who emphasized that soil testing is costly, slow, and logistically impossible to use for consistent measurement to drive meaningful progress. These voices became our blueprint. So we created Averra, a first-of-its-kind biosensor-based soil monitoring platform to revolutionize precision agriculture.
Now, I'm sure everybody here knows what a wearable device is. These are technologies that track people's temperature, heart rate, and sleep. They turn bodies' biological signals into insights to better people's health. Now imagine this same intelligence, but instead of the human body, it's being applied to the Earth's body. That's Averra. We turn soil biology into actionable insights for farmers and agronomists alike. At the heart of our technology are bioengineered biosensors, capable of detecting bioavailable plant nutrients in soil. Not only is this technology cost-effective, but it is also highly customizable, as it is able to detect a wide array of nutrients in soil.
The global soil testing market represents a massive opportunity. In the US alone, we can reach over 200,000 farms through partnerships with carbon and regenerative programs, allowing us to connect with farms and seamlessly integrate with existing systems to lower adoption barriers. And by promoting sustainable management, we can cut costs and overuse inputs and protect the farms that are critical to food system and climate resilience.
Our platform is set to unlock value across various markets. Averra closes a bridge that has been standing way too long in the soil testing market. We deliver on-site, real-time soil diagnostics. As we continue to validate and expand our technology, we aim to make key partnerships with leaders, industry leaders that share our same vision of data-driven agriculture.
We began in May of 2024 with nothing more than a scientific concept. Within just a few months, we built and validated our first benchtop prototype and are now working to scale towards early manufacturing readiness.
We're a team of scientists and engineers that have a vision to give soil a voice. Because when we understand soil, we unlock the potential to grow a better world. Thank you.